Auxin is one of the most important phytohormone and it is the first discovered phytohormone. It is synthesized from tryptophan in the tips of the plants and it regulate or induce the apical meristem. The auxin family includes many compounds, more than 200 auxin analogs are discovered till date, but some of the important auxin includes naphthalene acetic acid, indole 3 acetic acid, indole 3 butyric acid, and phenyl acetic acid. Among these, indole 3 acetic acid is the most studied and most important also. The function of auxins are it promotes stem elongation, induce adventitious root development, inhibit auxiliary bud development, it stimulate stringolactone synthesis, which further inhibit the lateral bud growth. Okay, and it regulate leaf abscission. So there are two similar words, leaf abscission and senescence, which are not exactly the same. Leaf abscission is normally the detachment of dead leaves or ripe fruits, but leaf senescence is more related with the age. It the same thing happens with the senescence also, but this is more related with the age that is it is due to loss of power of division and growth. And there are some chemicals which inhibit the oxygen transport. You can also remember it for exam, like a phytotrophin, TIBA, NPA, and again, most important thing also which are widely asked in exam, like what if oxygen increased and decreases? When oxygen increases, it induces xylem, and when oxygen is in low concentration, it induces phloem differentiation. Okay. Next, the structure and synthesis of auxin. Its structure is unsaturated, planar, aromatic ring, and it has a carboxylic side chain. Now it is synthesized in the shoot apical meristem, in young leaves, developing fruits, and seeds. If we go to the biosynthesis, it is synthesized from tryptophan. Tryptophan is denoted by W. Deamination of tryptophan leads to indole 3 pyruvic acid with the help of transaminase. Then decarboxylation of indole 3 pyruvic acid yields indole 3 acetaldehyde in the presence of decarboxylase. And finally, the oxidation of indole 3 acetaldehyde yields indole 3 acetic acid in presence of dehydrogenase. Now, before going to the signaling of auxin, this ubiquitination steps is very important to understand. It is important for both plants and animal. So, how it actually happens? The ubiquitin first get attached with the E1 protein with the help of ATP. Then this E1 uh, shifts this ubiquitins to E2. Then E2 gets attached with E3. E3 has dual sites. It attached to E2 and the target at the same time. So finally the E2 transfer its ubiquitin to the target and the proteasomal degradation of target takes place. Now the signaling of auxin. When auxin is not present, the AUX or IAA protein is bind with the ARF. ARF is auxin responsive factor and it inhibits its response. ARF is its receptor, so there is no expression of auxin response. Sorry, ARF is not the receptor, it is its a responsive factor. Then when auxin is present, it binds with TIR, which is its receptor. TIR is transport inhibitor response. It is also F-box protein or a component of SCF E3 ubiquitin ligase, which helps to ubiquitinate OX or IAA and finally leads to the ubiquitin mediated proteasomal degradation of OX and IAA, which helps to free the ARFs and we can finally see the expression of auxin responsive gene. And some of the physiological responses include cell elongation, cell differentiation, rooting, apical dominance, fruit set, and tropical responses also. Thank you.